Hello there, and welcome to this Biology Mind A-Level tutorial on transport of oxygen. So in the last lesson we learned about tissue fluid and the process which it is formed through, and we also had a little introduction to what the lymphatic system is. And in this tutorial we're going to come and have a look on the oxygen carrying capabilities of haemoglobin. So let's kick off because this is quite a chunky tutorial. So the haemoglobins are a group of chemically similar molecules found in different organisms. So there are different types of haemoglobins. There isn't just one type of haemoglobin, and we'll explore a different type of haemoglobin later on in our tutorial. So haemoglobin has 4 polypeptide chains. Now, the fact that it is composed of more than one polypeptide chain really defines it as having a quaternary structure. And remember, we learned about proteins in a previous tutorial and about how they have different structures: the primary, secondary, tertiary and quaternary structure. So the fact that the haemoglobin has polypeptide chains interacting with each other defines it as a protein that has a quaternary structure. Now, haemoglobin is composed of 2 alpha polypeptide chains and 2 beta polypeptide chains. And each of these polypeptide chains are associated with a heme group. So that means that there's 4 heme groups. And within each heme group, there's an Fe2+ ion. And it is this ion that oxygen associates with. So if there's 4 of these, that means haemoglobin can carry up to 4 oxygen molecules. Let's think about what haemoglobin actually does. So it's found in our red blood cells. Another fancy word for red blood cells, if you want to use it, is erythrocytes. And haemoglobin allows red blood cells to transport oxygen from the lungs to the whole body. And when combined with oxygen, we call haemoglobin oxyhaemoglobin. So note this oxy stem, which means that it's haemoglobin combined with oxygen. Now let's have a little think about oxygen transport by haemoglobin and have a think about some key terms that you will need to get very familiar with by the end of this tutorial, and we'll be using them lots and lots, so don't worry, I'm sure they'll come very naturally by the end. So number 1 is dissociation. So this is the loading of oxygen onto haemoglobin. So we say that oxygen associates with haemoglobin, it binds with haemoglobin. Likewise, there's dissociation, which is the opposite of this. So when oxygen is unloaded from haemoglobin, we say it's dissociated from haemoglobin. Now, affinity is the degree to which one substance combines with another. So we say that haemoglobin has either a high or a low affinity for oxygen. And haemoglobin can change its affinity for oxygen. So when oxygen concentration, these square brackets around the oxygen symbol refers to concentration. So when the oxygen concentration is high, haemoglobin has a high affinity for oxygen. And the opposite is true as well. So when oxygen concentration is low, haemoglobin has a low affinity for oxygen. Now, this all makes a whole lot of sense when you think about it, because in the alveoli, where oxygen concentration is very high, we want haemoglobin to bind to oxygen. So therefore it makes sense that haemoglobin has a higher affinity for oxygen. Then vice versa, in the respiring tissues where oxygen concentration is low, haemoglobin has a lower affinity for oxygen, which is good because we want haemoglobin to give up its oxygen to the respiring cells and tissues. So that's a really neat way to think about how the affinity of haemoglobin changes for oxygen. Now let's have a think about our different terminologies when it comes to oxygen. So partial pressure of oxygen is a measure of the oxygen concentration. So a high partial pressure of oxygen corresponds to a high concentration of oxygen, and vice versa, a low partial pressure of oxygen corresponds to a low oxygen concentration. And haemoglobin has different affinities for oxygen depending on its partial pressure. So remember, as I said on the last slide, respiration uses up oxygen, so the partial pressures of oxygen are lower in respiring tissues, and vice versa. In the alveoli, where the oxygen concentration is a lot higher, the partial pressure of oxygen is higher. And remember, it's here in the alveoli that haemoglobin has a higher affinity for oxygen. So let's go on to have a look at an oxyhemoglobin dissociation curve where we can make a bit more sense of this. So we describe these curves as sigmoid shaped, which just means it has this S structure, as you can see here. So for the first part of us going through the slide, I just want you to have a think about our adult hemoglobin first of all, which we call hemoglobin, so HbA for adults. And it's this orange curve here. Ignore the blue curve for now, we'll talk about that in a few minutes. So first of all, have a look at it and have a look at how the gradients curve is initially shallow, then it steepens, and then it flattens and levels off. But what does this all mean? So to figure out what it means, we need to have a think about what our axes are first of all. So on the x-axis is our partial pressure of oxygen. So the higher up our x-axis we go, the higher the partial pressure of oxygen. On the y-axis is our oxygen saturation of haemoglobin. So the higher up our y-axis we go, the higher the saturation of haemoglobin. Now, what we can see is that at lower partial pressures of oxygen, our oxygen saturation of haemoglobin is lower. At higher partial pressures of oxygen, the oxygen saturation of haemoglobin is higher, with this very steep change in the middle. So this is a really important property of haemoglobin. It's what we call the cooperative nature of oxygen binding to haemoglobin. So all this means is, as oxygen molecules number 2 and number 3 bind to haemoglobin, haemoglobin changes its 3D structure. It changes its structure to make it easier for oxygen to bind, which means that the more molecules of oxygen we bind to haemoglobin, the more haemoglobin changes its structure in order to make the subsequent molecule of oxygen bind easier. So this is really important because it means that we get this lovely steep rise here in our curve. Now, going back to points number 1 and number 3, so our gradients curve is initially shallow, so at low partial pressures of oxygen. So I'm going to write it as this actually, so at low concentrations of oxygen or as at low partial pressures of oxygen, for example, in our respiring tissues, our oxygen saturation of haemoglobin is lower. Now, at our higher partial pressures of oxygen, our oxygen saturation of haemoglobin is higher. So remember, this is in the alveoli, whereas the lower partial pressures of oxygen are found in respiring tissues. So what we say is in respiring tissues, haemoglobin has a lower affinity for oxygen, which means that it dissociates from oxygen. Whereas here, haemoglobin has a higher affinity for oxygen, which means it associates with it. Which makes sense because remember in the alveoli we want haemoglobin to associate with oxygen, whereas in the respiring tissues we don't want it to. We want it to release oxygen to these respiring tissues. Now, we've had a talk about how this works in adult haemoglobin. So before we move on, actually, I just want to cover what happens in fetal haemoglobin. So fetal haemoglobin, we write as this, HB for haemoglobin and capital F for fetal. And it has a slightly different structure to adult haemoglobin in that it's formed from 2 alpha polypeptides and 2 gamma polypeptides, whereas adult haemoglobin is formed from 2 alpha polypeptides and 2 beta polypeptides. So this is our structure of fetal haemoglobin, and this has a slightly different structure to adult haemoglobin, and you can see on this curve, so the blue curve is the fetal haemoglobin, you can see it's different to the oxyhaemoglobin dissociation curve for adult haemoglobin. It's shifted to the left. Now, what does that actually mean? It means that fetal haemoglobin, so I'll write this up here, fetal haemoglobin has a higher affinity for oxygen. And we can see that when we look at this graph. So for example, at this partial pressure of oxygen of 26.8, adult haemoglobin is 50% saturated, whereas fetal haemoglobin is probably about 70% saturated. So fetal haemoglobin has this higher affinity for oxygen, which makes sense because remember the fetus is receiving its oxygen from the mother's placenta. The fetus doesn't use its own lungs when it's in the mum. So the mother's placenta receives blood from mum that's already passed through some of the body. So it's going to have a slightly lower partial pressure of oxygen than the blood in her alveoli. So the fetal haemoglobin in turn need to have a higher affinity for oxygen to make sure that it binds that oxygen, even though there's a little bit less of it in there. So it really does make sense that fetal haemoglobin needs to have a higher affinity for oxygen. Okay, so we've talked a bit about our adult haemoglobin, and we've talked a bit about our fetal haemoglobin. We understand that this is a sigmoid-shaped curve and that there's a cooperative nature of oxygen binding to haemoglobin, and that in the alveoli, where the partial pressure of oxygen is high, our haemoglobin is going to take on the oxygen. It has a high affinity for oxygen, it's going to associate with it. Whereas in the respiring tissues, where the partial pressure of oxygen is low, it's going to dissociate from oxygen and release that oxygen to the respiring tissues, which is what we want. So I always like to think to myself, if I ever forget this in an exam, I can kind of work it out from first principles, because I can kind of think, well, what do I want the haemoglobin to be doing in the alveoli? And what do I want it to be doing in the respiring tissues? And you'll find it actually does what you want it to do, which is quite smart. Okay, so we've had a look at our basic oxyhaemoglobin curve. So now we're going to have a look at something called the Bohr effect. Now, respiring cells produce CO2. So this increases the concentrations of CO2 around respiring cells. And carbon dioxide dissolves in water in blood to form carbonic acid, so something acidic. And carbonic acid in turn causes a slight change in the shape of haemoglobin. So it causes this change in the shape of haemoglobin, which actually shifts the curve to the right. So you can see here that at a higher concentration of carbon dioxide, the curve is shifted to the right compared to here, which means that if I just trace up at a partial pressure of oxygen, so it means that if we go from this partial pressure of oxygen of 20, our oxygen saturation in our higher carbon dioxide concentration is about, let's say, 17. Well, actually, no, that looks more like a 15 to me. 15% oxygen saturation of haemoglobin. Whereas if we trace it up to our much lower concentration of haemoglobin, we can see that our oxygen saturation of haemoglobin is about 85%. So you can see that's a huge difference, isn't it? Now, that's because haemoglobin has a lower affinity for oxygen at higher partial pressures of carbon dioxide, causing oxygen to be released, which makes sense. Again, you can work it out from first principles, because in our respiring cells, we have our higher concentrations of carbon dioxide. Therefore, we want our haemoglobin to have a lower affinity for oxygen so that it releases oxygen, and vice versa. So in our alveoli, we're going to have lower partial pressures of carbon dioxide, so we're going to be on this curve, and our Bohr effect is going to shift our oxyhaemoglobin dissociation curve back to the left, so back towards this lower concentrations, And that's going to mean that haemoglobin has a higher affinity for oxygen, causing oxygen to be taken on to associate to haemoglobin, which again is what we want in the alveoli. So this effect of carbon dioxide concentration on the affinity of haemoglobin for oxygen is known as the Bohr effect. And remember, higher carbon dioxide concentrations cause the curve to shift to the right, and that's because of this change in shape of the haemoglobin that's caused by the high carbon dioxide levels. Okay, so we've talked about our haemoglobin in our adult, our haemoglobin in our fetus, and we've talked about the effect of different concentrations of carbon dioxide on our haemoglobin. So now we're going to think about haemoglobin in different organisms. So we've already met one different type of haemoglobin. Remember, we met our adult haemoglobin, which had the structure alpha 2, beta 2, so 2 alpha polypeptide chains and 2 beta polypeptide chains. And we also met our fetal haemoglobin, which had the structure alpha 2, so 2 alpha polypeptide chains and 2 gamma polypeptide chains. And this property of fetal haemoglobin meant that the fetal Oxyhaemoglobin dissociation curve, gosh, what a lot of words, was shifted over to the left, which meant that fetal haemoglobin had a higher affinity for oxygen than adult haemoglobin. Now, in the same way that this occurs between fetus and adults, different species have different types of haemoglobin. So this allows them to adapt to the environment. So I'm going to draw out a graph here to just show this. So this is our y-axis, and again we've got our percentage saturation of haemoglobin by oxygen. And then, so this is our y-axis, sorry, did I just say x? I meant y. And then this is our x-axis, which is our partial pressure of oxygen. So the higher up our x-axis we go, the higher the partial pressure of oxygen, and the higher up our y-axis we go, the higher the percentage saturation of haemoglobin by oxygen. So if I just draw a normal adult haemoglobin dissociation curve, so let's just label this as human haemoglobin, or let's label it as HbA now that you know that terminology. So that's our adult haemoglobin. So now if I draw this curve, I want you to have a think about what this curve is. So this curve is showing that at higher partial pressures of oxygen, our haemoglobin has a higher affinity for oxygen. So this curve actually represents a species of animal that is adapted to living in low oxygen environments. And we can tell this because at lower partial pressures of oxygen, the haemoglobin is binding more oxygen, it's associating with more oxygen. That's really interesting, right? And vice versa is also true. So if I drew this curve, so this is a dissociation curve representative of a metabolically active animal living in an oxygen-rich environment. And we can tell this because at lower partial pressures, or I should say, at higher partial pressures of oxygen, we're still getting our unloading of oxygen to our respiring tissues. So there's a lower affinity of haemoglobin for oxygen in this animal compared with this animal which lives in the oxygen low environment. So that's an example of how different species might have different types of haemoglobin in order to adapt to their environment. And just to doubly get that in, so species living in environments with low oxygen concentrations will have haemoglobin with a higher affinity for oxygen, and species living in environments with high oxygen concentrations will have haemoglobin with a lower affinity for oxygen. Okay, so that has been quite an exhaustive tutorial. We've covered a lot of things. I really recommend you get your head around those oxyhaemoglobin dissociation curves, and as I said, try to contextualize it. Try to think, well, Where am I getting low partial pressures of oxygen from? An example of that would be in respiring tissues, and therefore what do I want my haemoglobin to do? Well, I want it to give up that oxygen, I want it to give it to the respiring tissues. And that can help you figure out yourself exactly how haemoglobin is reacting to the differing environments that it's in. So I hope that makes a whole lot of sense to you, and I will see you for the next tutorial.